Welcome to Chapter 5, where the innate immune system finally meets the adaptive immune system. So we've sort of built it up so far. We've talked about what our first response is when a pathogen enters the host. We are now going to talk about where these two very important arms of the immune system meet for the successful elimination of pathogen. In order for the adaptive immune system to finally become activated, within a few hours of the initiation of the acute inflammatory response, your antigen-presenting cells that have played a really important role in innate immunity begin to leave the inflamed tissue and head to the draining lymph nodes. These could be dendritic cells, which are probably the most efficient of these APCs. And they will actually retract up their membranous processes to begin the journey to the draining lymph node. So let's just take a moment to review where we've come from so we know sort of where we're going. Dendritic cells and macrophages bind to antigens, as we talked about, through these pattern recognition receptors with a pretty limited diversity. Activation through those receptors helps to induce an acute inflammatory response in the tissue, leading to the production of the pro-inflammatory cytokines. These cytokines cause a phenotypic change in the phagocyte, which helps to alter their migration pattern and enhance their function. So let's take a look at what we just talked about, but sort of in a diagrammatic form. Let's look at the top where you can see a random site of infection. You can see an entering microbe, and what is likely happening during this infection is that we have acute inflammation happening. What we are about to look at is we are going to follow that antigen presenting cell from that site of acute inflammation to the draining lymph node, where we can now initiate the adaptive immune response. We are going to sort of walk through this eventually as it happens through the lymph node, but just keep in mind that antigens that get into the blood will eventually go to the spleen, where we will be having a similar response as far as the cell-cell interactions. Considering how many pathogens that enter the body on any given day, it seriously would be almost impossible for the lymphoid cells to travel to all of those sites to protect the host. Therefore, the lymphocytes are constantly recirculating and going to the secondary lymphoid tissue, where they're most likely to come in contact with that specific antigen. Dendritic cells, as we just talked about, are going to start their migration from the acute site of inflammation to that secondary lymphoid tissue by expressing a chemokine receptor called CCR7. CCR7 is activated by chemokines, and those chemokines will bind to CCR7, allowing them to exit the tissue and begin that migration to the secondary lymphoid tissue. Once the dendritic cells become activated through CCR7, they switch their focus from capturing antigen to presenting antigen. The activated dendritic cells then concentrate in those draining lymph nodes, becoming trapped in the paracortex, which we're going to take a look at in just a few minutes, where you'll be able to see that the dendritic cells are then going to present that antigen to the T cells that reside within the paracortex. Naive T cells also express CCR7, bind to chemokines on high endothelial venules within the secondary lymphoid tissues, and also migrate to the paracortex. As we mentioned, if the initial tissue damage is sufficient, the cells can also be flushed into blood vessels, ultimately becoming trapped in those vascular sinusoids of the spleen, and we would have a very similar reaction in the spleen. The secondary lymphoid tissues, including both the lymph nodes and the spleen, are the sites where naive B cells and T cells will first be exposed to their specific antigens. And what you want to think about are that these naive B cells and T cells have sort of a short period of time to come in contact with antigen that they're specific for, or they will eventually die off. So they are very focused on recirculating through the secondary lymphoid tissue in the hopes of finding their specific antigen. Let's just take a look at the structure of a lymph node. Lymph nodes are small aggregates of secondary lymphoid tissue with extremely organized structure that are found along the lymphatic channels in the body. You'll notice there are multiple afferent lymphatic vessels and only one efferent lymphatic vessel to ensure sort of that the lymph flows in one direction. You'll notice that the lymph node is very compartmentalized, with the outer cortex being predominantly where your B cells reside. Right next to the cortex is the paracortex, where we're going to find most of our T cells. And then we find the medullary area, where we are going to find sort of our activated cells and our memory cells prior to their descent into the lymphatics to begin to protect the rest of the body. Lymph nodes in general drain most of the tissues, drain certain aspects of the mucosa, and are designed to initiate immune responses to antigens that get into either the mucosa or the tissue. In addition to the B cells and the T cells, it's also sort of important to know where the antigen-presenting cells other than B cells live within the secondary lymphoid tissue. If we take a look at the dendritic cells, you can say that those reside mostly within the T cell areas of the lymph nodes and the spleen. And that makes perfect sense because remember, these are the cells that have captured the antigen and now are attempting to present it to T cells within the paracortex. As we look at the macrophages, we can see that the macrophages sort of live all throughout that secondary lymphoid tissue, both within the cortex and the paracortex. The spleen is the other major secondary lymphoid tissue that is extremely compartmentalized, and it really handles the immune responses to blood-borne antigens. The splenic artery that enters the spleen is surrounded by cuffs of lymphocytes, and this is known as the periarteriolar lymphoid sheath, and that is the T cell specific area of the spleen. This is a really good image of the spleen because it sort of shows two different views of what's happening. Let's take a look at the longitudinal section first, because you can easily see the splenic artery, and you can see sort of the purple area around the splenic artery, which is the periarteriolar lymphoid sheath, which we already said was predominantly T cells. Okay? We can also see sort of the gray area surrounding that Powell's region, which are the marginal zones, which is predominantly where our B cells are going to reside. Now, one thing that's important to note, and this is important in both the lymph nodes and the spleen, is that during an immune response, you can have a highly organized structure within the lymph node and the spleen called the germinal center. And the germinal center is essentially just rings and rings and rings of proliferating lymphocytes. And there are lots of important events that happen within the germinal center that are going to be important later on. One of them is isotype switching or class switching, that we will talk about a little bit more when we get into humoral immunity. Another is somatic hypermutation, which also happens in the chapter on humoral immunity. And the other sort of important thing that happens within the germinal center is sort of the decision of a B cell to become either a memory B cell or a plasma cell. And again, we'll talk about that a little bit more when we get to humoral immunity. But very important events happen within the germinal center. And it's important to know that even though you see a germinal center within this image, and there is also one located in the image or the figure of the lymph node, that germinal centers are really only going to be present during an active sort of immune response. Now let's take a look at the transverse section of the spleen, where again, you can see the central arteriole. You can see that surrounded by the Powell's region, which again is mostly T cells. Again, surrounded by the marginal zone of B cells. And right at the interface, sort of between those, we have the germinal center. The next step in the story of the innate immune system meets the adaptive immune system is antigen processing and presentation. So recall, Dr. Ali already talked to you a little bit about the MHC. She overviewed for you what class 1 MHC looks like, what cells class 1 MHC is expressed on, and also class 2. So we are first going to talk about the exogenous pathway of MHC loading, which deals with how antigens are going to be presented on class 2 MHC molecules. This is sort of the first step in the elicitation of the adaptive immune response to a primary challenge. Although some small, easily digestible antigens are almost totally degraded and exocytosed by phagocytes, we do have parts of those molecules that are obviously processed and presented onto these MHC molecules. In the exogenous pathway of MHC processing and presentation, we first need to talk about antigen-presenting cells. Under duress, lots of different cells can express class 2 MHC, but the ones that you really need to know for step 1 are the professional APCs, which include dendritic cells, macrophages, and B cells. It is the responsibility of these cells to process antigen and then load the antigen into the groove of the class 2 MHC molecule. These antigen-presenting cells have slightly different functions that help to elicit different sorts of immune responses that are required to eliminate different types of pathogens. Let's take a look at some of the antigen-presenting cells that are important in antigen processing and presentation. Dendritic cells are sort of the primary APC that we think about when we're talking about activating naive T cells, simply because they constitutively express the markers that are required for this sort of activation. Macrophages are really going to be essential in helping to activate T helper cells and helping us to elicit a cell-mediated immune response. B cells are sort of unique in the APC arena because they're actually the only APC that specifically recognizes antigens. And of course, this happens through the B cell receptors. This table helps to summarize the different antigen-presenting cells, looking at the different co-stimulatory molecules that they express, their expression of class 2 MHC, and sort of the major function. Some of the co-stimulatory molecules that we're going to be talking about in upcoming chapters include B7, which binds to CD28 on T cells, and also CD40, which binds to CD40 ligand on T cells. So those are known as co-stimulatory molecules, and those are going to become much more important when we talk about activation of cells in the next chapter. The expression of class 2 MHC in some of these cells are constitutive, but under the influence of interferon, particularly interferon gamma, can be upregulated. And you can take a look then at some of the major functions of those antigen-presenting cells within the immune response. As we said, we're first going to discuss the exogenous pathway of MHC loading, which handles threats that are extracellular in nature. Bacteria and other pathogens that are phagocytosed by these antigen-presenting cells are killed and loaded onto a class 2 MHC molecule for presentation to a T helper cell. So let's take a look at that pathway of antigen presentation. So let's walk through the exogenous pathway for antigen presentation. As you look at this figure, there are a few things we need to talk about before we can really talk through the process. Number one, keep in mind that the MHC is located within the endoplasmic reticulum. So recall that in addition to class 1 MHC being within the endoplasmic reticulum and being responsible for presentation of endogenous peptides, the class 2 molecule is also located within the endoplasmic reticulum. So keep that in mind. Now, take a look at the left side of this figure where you can see the antigen being taken up by this APC and processed into smaller peptides. Okay? What has to happen now is we need to get those peptides somehow loaded up on a class 2 molecule, which is still sort of located and situated within the endoplasmic reticulum. What I want you to notice, though, as you look at that class 2 MHC molecule in the ER, you can see that its peptide binding cleft is actually locked up or bound with a peptide known as the invariant chain. Therefore, when endogenous peptides get into the endoplasmic reticulum, in the case of perhaps a viral infection, they get sorted properly and essentially shuttled into the class 1 molecule, because while the class 2 MHC is in the ER, it actually is not able to bind peptides. So that's sort of an important point. Okay? Now, the class 2 MHC will move through intracellular trafficking from the ER through the Golgi and eventually fuse with the phagolysosome. So you get one big happy vesicle. So notice that we have the exogenous peptides within that large vesicle. We also have the class 2 MHC molecule, along with another molecule that I know Dr. Ali introduced to you earlier, which is the HLA-DM. Now, what we're going to see within this vesicle is an exchange. We're going to see an exchange of the invariant chain for these exogenous proteins, and this is important, if the exogenous protein actually has a higher affinity for that class 2 molecule than the invariant chain. And the HLA-DM sort of facilitates that exchange, and the invariant chain is released and replaced with exogenous peptide. Once that exchange happens, you can see that the class 2 MHC with exogenous peptide is then fused to the cytoplasmic membrane so that that peptide can be presented to a CD4 positive T helper cell. Of importance to note, if the class 2 molecule does not find a peptide that it can bind with a higher affinity than the clip, that empty class 2 molecule is unstable, it gets degraded, and never makes it to the cell surface. The endogenous pathway of antigen processing handles threats that are intracellular. So in particular, we're really talking about viruses, potential transformed cells. Intracellular proteins, regardless of sort of where they come from, are degraded and targeted by ubiquitin and then degraded in the proteasomes. And it's these peptides, regardless of sort of where they come from, that can be loaded up onto the class 1 molecule. So let's walk through the endogenous pathway of antigen presentation. We're going to use a virus as an example. Viruses, as we already know, are obligate intracellular. They reproduce within our host cells, so the viral peptides that are made are being made within the host cell cytoplasm. These peptides can be targeted for degradation by being tagged with ubiquitin, and they can head for the proteasome, which will degrade those proteins into smaller peptides. Now, that's sort of an important step because, as Dr. Ali talked about before, the class 1 MHC is actually only capable of binding really small peptides of about 8 to 10 amino acids. So a large viral protein would not be able to fit in that peptide binding groove. So it's important that they are processed into smaller peptides. Once those peptides are produced from degradation in the proteasome, we have another sort of barrier because the MHC molecules are actually located in the endoplasmic reticulum, but the peptides are located in the cytoplasm. That's where the TAP molecule becomes really important. TAP stands for transporter of antigen processing, and the way I want you to think about TAP is that it's like a molecular tunnel that actively takes peptides from the cytoplasm and loads them up onto a class 1 MHC molecule. You can see there is a molecule called tapesin here that sort of bridges the class 1 MHC to the TAP transporter to easily facilitate the addition of that peptide onto a class 1 MHC molecule. Class 1 is notoriously bad at being folded properly. It requires that beta-2 microglobulin that Dr. Ali talked about earlier, but it also likes to have peptide within that peptide binding groove in order to be in the proper conformation to actually present to cytotoxic T cells. Once that peptide is actually present within the class 1 peptide binding groove, essentially that class 1 peptide complex will move through the cell, will go through the Golgi, and eventually be in a vesicle that fuses to the cytoplasmic membrane for presentation to a cytotoxic T cell. Now, it is the CD8 of that cytotoxic T cell that is actually recognizing that class 1 MHC molecule, so we say that that is restricted to class 1 MHC. This pathway, a little bit more high yield for step 1, and I'm going to tell you why in just a minute. Not that the exogenous pathway isn't as important. This is the one that tends to make its way into question pools a little bit more, and I'll explain why. When you get questions on USMLE, a lot of the times they're going to phrase the question in forms of the context of somebody running an experiment where they monkey around with a gene, they delete a gene, and they want you to know what the outcome is. And it's very easy to do that here because there are a lot of molecules that come into play that if you don't remember they have something to do with class 1, it's going to be hard to answer those questions. For example, if we had a mutation where TAP was mutated, you can just automatically sort of envision what's going to happen, right? So the peptides aren't going to be able to get onto that class 1 MHC molecule. Class 1 is not going to fold properly, and class 1 is not going to be expressed. So essentially, if you have a TAP mutation, you also have a class 1 MHC, essentially, mutation where your patients aren't going to be expressing class 1 MHC. That's not the whole story though, because as Dr. Ali told you about when she was talking about positive selection, in order for a cell to be positively selected, it actually has to interact with either class 1 or class 2 MHC. If there is no class 1 MHC, we cannot get the positive selection of CD8 positive T cells. So the downstream effect of having a TAP mutation is that you have a class 1 MHC deficiency and you wind up having a deficiency of CD8 positive T cells. Now, the reason, again, that they like to ask those questions a little bit more frequently than issues with the class 2 pathway is within the class 2 MHC pathway, they have to ask those questions more specific to class 2. So you would sort of be given clues that you had a mutation in class 2 versus just saying you had somebody in a lab that monkeyed around with TAP and you'd have to figure out what that meant, right? So you have to know that TAP mutations result in class 1 deficiencies, result in CD8 deficiencies, and in general, what they're likely to ask you is what that means. What are you going to see in a patient, or if they're doing research in an animal? You're going to see an increased susceptibility to viral infections. We discussed the importance of the proteasome in the endogenous pathway of antigen presentation. So now let's take a look at what happens if we can actually inhibit the function of the proteasome. We wouldn't want to do that normally, but consider that oncogenic cells divide at a pretty high rate. That requires a much higher rate of protein synthesis than most cells. Normal cellular proteins are also degraded through the ubiquitin pathway. And if we inhibit the proteasome, we can actually induce apoptosis in certain tumor cells by interfering with the degradation of some of the regulatory proteins that are going to be important in maintaining cellular homeostasis. At the publication of this book, there were only 2 drugs that were actually FDA approved that were proteasome inhibitors. Probably there are already more now because that's what happens in both immunology and pharmacology. Both were FDA approved to treat multiple myeloma. They are looking at other trials for leukemias and lymphomas for these proteasome inhibitors as well. The last concept we're going to talk about in Chapter 5 is something called cross-presentation or cross-priming. It is really sort of a cool thing where dendritic cells have a role in not only presenting antigens on a class 2 molecule to a CD4 positive T cell, but can also present antigens to CD8 positive T cells in this process. This is going to be an important process in understanding what Dr. Alley is going to sort of talk to you about in some of the later chapters when we talk about the importance of interleukin-2 being produced from a T helper cell that also helps to activate a cytotoxic T cell. In cross-priming, dendritic cells, which we have already discussed as professional antigen-presenting cells, can actually phagocytose an entire viral infected cell. And through that can actually activate and prime not only CD4 positive T cells through class 2 MHC, but can prime at the same time CD8 positive T cells through class 2 MHC. And as we talked about earlier, this is going to be an important concept when we talk about cytotoxic T cells becoming activated killer cells through the production of IL-2 from the T helper cell.